Hello students, this is Biobotany class by Mini. Today's our topic is life cycle of Ustilago. In this case, you can see that is the infected plant, that is Ustilago. And Ustilago means, first you should understand what is Ustilago is a smut fungi. It means where Ustilago, the commonly 300 species of Ustilago is called smut fungi, where and these mostly crop will be wheat, corn, sugarcane and some grasses which are present in the parasitic form. And this smut disease which caused smut diseases, and these plants grains form into black powdery form, which is Ustilago. It means Ustilago, it means smut fungi. Smut fungi. Okay, so now description the life cycle of Ustilago. In this case, first it formed chlamydospores. Chlamydospore, which have two layering. Outer layer is exine, inner is intine. So chlamydospore disseminated by wind and the sorus are formed. It is infected spike, and when the disseminated start by wind and the sorus will be free. And so what is the question? What is sorus? Sorus is the chlamydospore, binucleate chlamydospore. Binucleate chlamydospore. Binucleate chlamydospore. So after free from the sorus, that become that fall into the ground or into mix in the soil with grains, and their viability will be 5 to 6 months. So before germination, binucleate chlamydospore fuse with diploid nucleus. Binucleate chlamydospores fuse with diploid nucleus, and during germination spore water absorb during germination spore absorb water and it swell. After swelling, the exine layer will be burst, and intine layer formed a germ tube, that is germ tube. And in this case, which is called, after germinating germ tube, it form promycelium, which is called promycelium. This germ tube is called promycelium or basidium. Basidium, D-I-U-M. Basidium, promycelium or basidium. In this stage, chlamydospores diploid nucleus reduction division starts, and it formed 4 haploid nuclei. These are 4 further it divided, it formed 4 haploid nuclei, and after reduction division make 4 haploid basidium cells. It means each single haploids have 1 single nuclei, and after that the cell divide into 2 daughter cells. At the cell divider, that case after the multiplication of basidiospores by the process of budding. This process is called budding process. And basidiospore will be thin-walled, uninucleate, oval structure. Thin-walled, uninucleate, there have single nuclei, oval structure, and by the process of budding they multiplicate. So in the germination of basidiospore, how it will start? The germination of start through the monokaryotic mycelium, which will be a positive strain and the negative strain. That is the life cycle of Ustilago. In quick view, now understand the cell wall of basidium chlamydospore, the cell wall, then the infected sorus fall down into the and mix into the soil with grain, and before germination contact with the water and the cell swell. After swelling, the outer wall will be burst. Outer layer, it means exine burst, and intine cell will be formed a germinative tube. This germinative tube is called promycelium. Germ tube or germinating tube. This promycelium is called basidium. And these basidium cell further divide into 4 haploid cell. Each haploid cell have individual nucleus, and it is binucleated, and after that they replicate, they multiplicate by the process of budding, and therefore each individual basidiospore, it means they formed 4 basidiospores cells. And these how the basidiospore cell, the basidiospores are thin walled, uninucleate and oval structures. Okay, and how it is formed? It is forming by the process of budding. And how the basidiospore germinate? The haploid basidiospore germinate with the monokaryotic mycelium, and these mycelium will be positive and negative strain.